Dear ladies and gentlemen, I would like to present you the results of the research that is devoted to genetic basis of ischemic stroke made with the genome-wide association study and machine learning approaches. The ischemic stroke is a neurological deficit of sudden onset due to brain infarction. It happens because of blood clot or plaque formation in blood vessels. They decrease the content of nutrients and oxygen, resulting in the loss of function by neurons and their death. The ischemic stroke is the second cause of death all over the world. In Russia, there are about 500 cases per 100,000 people per year, and the mortality from ischemic stroke is 175 cases per 100,000 people per year. The ischemic stroke has genetic basis, and there about 1% of ischemic stroke that was resulted due to monogenic disorders, and these are Cadecial, Carecial, Fabry disease and other syndromes. There are about 15 different syndromes that has monogenic origin, and they result in ischemic stroke. But we were interested in more common cases. They are polygenic, and at the moment there are several genes candidate that are associated with the ischemic stroke, and one of them were obtained with PCR methods and the others with genome-wide association studies. And that's clear that we need more genome-wide association studies because this list is not accomplished. And to do this, there is a very nice opportunity. The Department of Molecular Basis of Human Genetics and Pirogov Russian National Research Medical University created a collections of blood of patients with ischemic stroke, and there are more than 1,000 cases in this collection. And moreover, they genotyped them, and also there is a collection of people that can be used as a control group, and they're so-called population control. And using this data, we decided to run genome-wide association study and also apply the so-called advanced statistical approaches as machine learning. But there are some peculiarities in the data. In this picture, you see the distribution of age in case and control groups. And that's clear that average age in control group is a little bit different than average age in cases, and that's a problem. We need to do something with this, and in this research we take into account this difference. And another peculiarity has to do with the fact that the data were genotyped using different type of microarrays. On the left graph we see the number of markers for each type of microarrays. There were 5 types. And on the right graph we see the number of individuals genotyped with each type of microarrays. And the peculiarity is in that the largest amount of people were genotyped with the microarray that has the less number of markers, about 300,000 markers. So, to run GWAS and machine learning, we need to merge these data sets, and they are made with different number of markers. And merging of dataset was a big challenge of this research, and in this slide I would like to outline 3 different strategies that were applied: Protocol 1, Protocol 2 and Protocol 3. So the first idea was just to let's merge all datasets and then run quality control procedures, then we needed to convert from genome assembly 18 to genome assembly 19 and run genome-wide association study in machine learning. But when we merged all data set, we've got this problem. These are the histogram of SNP missingness. On the x-axis we see the percent of missing genotypes per each SNP, and there are definitely some patterns of missingness. And moreover, if we just apply this threshold, let's missingness to be just about 10% or something like this, we've got about 85,000 markers. So, but the patterns were a huge problem, because finally it happened that they influence the results of machine learning. And that's why we decided to apply different protocols. And the idea was in that let's firstly merge the data by microarray type. We have 5 types of microarray, and within each type we merge firstly the data. Then we apply quality control for each type of merged data. And after that, we made one more step. We created the protocol number 3, where we imputed the missing values of genotypes, because we wanted to be sure 100% that they will not influence the results of machine learning and genome-wide association study. So, let me say a few words about the quality control, and we were interested only in ischemic stroke. The original collections of blood, they had not only ischemic stroke but also hemorrhagic stroke, and we were interested only in single nucleotide polymorphism, and only autosomes were selected for the analysis. And we filtered the data by call rate for individual, and also we filtered the data by call rate for SNPs. In the case of the protocol 3, where we applied the imputation, the threshold value was a little bit less, because 30% of missing data, it's okay to impute these values. And also we encountered the problem that the intersection of sample IDs in different data sets, and we made sample ID to be unique. And of course we cared about the sample that should be unrelated. We use Blink and King program to check for relatedness and remove the related people by p hat more than 0.2. We also noted the presence of duplicated samples, and we removed them. And finally, we cared about the homogeneity of genotypes with the principal component analysis. Now let me show a couple of slides, and here we see the result of principal component analysis. The green dots are control group and the brown dots are test group, and you can easily see that there are some outliers here, and we removed them by just applying the cutoff of PC1 0.1, and we applied this cutoff. When we removed these individuals from test sample, we've got this PCA that looks a little bit nicer and more homogeneous. Now let's speak about the GWAS. So as the result of control quality procedures, we've got these numbers of SNPs in our 3 protocols, and this number of individuals in 3 protocols for case and control groups. And we tested different tests. We applied chi-squared test using Blink. We also applied Fisher test using SNP test tool, or with different models, additive, dominant, and recessive inheritance model. And we also applied Bayesian test using additive inheritance model. And when we tried to consolidate all the results, we noted that actually they all look similar. And I would like to show a couple of Manhattan plots that reflect this idea. And on Manhattan plot we have the chromosomes on the x-axis and a minus log of the base 10 of p-value on the y-axis, and we see some outliers here on the chromosome 4 and the outliers on chromosome 7, and the red line shows the p-value 10 to minus 8. And this is the result of a Fisher test. And now let's see the Manhattan plot made for the results obtained with the Bayesian test. And on the y-axis we have Bayesian factor, and just a very similar situation. We also see the outlier on chromosome 4, and there are some outliers on chromosome 7. And when we consolidated all this data for all three protocols, we noted that there are a couple of SNPs that are almost always present, and we took these SNPs as to be associated with the ischemic stroke. And then we were interested about the location of the SNPs on the genome. And speaking about the first SNP, it's happened that it's located in the intron of the gene UNC5C, in the first intron of the gene. And this gene is very interesting. It codes the proteins that is called nitrins, and nitrins direct exon extension and cell migration during neural development. And moreover, this gene is highly expressed in brain. This is very nice. And speaking about the second candidate, SNP, it's intergenic. It lies between the gene DDX43P2, but the closest gene is so-called von Willebrand factor C domain containing 2, and the distance to this gene is about 156 kilobases. And we're not sure whether this polymorphism influences this gene or not, but that's interesting that this gene encodes a bone morphogenic protein antagonist, which is possibly involved in neural function and development and may have a role in cell adhesion. Now let's speak about the ischemic stroke as to be related to other disease. It's happened that there's some relation between ischemic stroke and other diseases. And we were surprised when we found that the gene that was identified by us as possibly being associated with ischemic stroke, and the gene is UNC5C, is well known that this gene is associated to Alzheimer's disease. And if we go to the resource called Malacards and then choose related diseases for Alzheimer's disease, we've got this table, and with a very high score we see that ischemic stroke Alzheimer's disease has already at least 4 genes in common. So probably the UNC5C will be added to this list as common to ischemic stroke and Alzheimer's disease. And speaking about the second SNP we identified, it's happened that this SNP is associated to Parkinson's disease. And moreover, it's well known that there are 5 genes in common that are associated to both ischemic stroke and Parkinson's disease. This is very interesting that a couple of SNPs we identified, they have something in common with other neurological disorders. And probably all of these disorders have common molecular mechanisms. Now, just let me tell you about the results of application machine learning approaches. And in this table, we have in columns the names of different machine learning approaches we applied. These are logistic regression, support vector machine, K-neighbors, random forest, XGBoost. And in columns, we have different modifications. So this column contains the results of application, the matrix that is called area under receiver operating characteristic curve, but in this column we have the dataset that was a little bit transformed. The main problem in applying machine learning approaches is the problem of size. We have 85,000 markers, attributes, and only a few hundred individuals. So we need to decrease the size of attributes, and that's why we apply a different way of doing this. And one of the way was singular value decomposition. Then we also tried diode approach and UMAP approach with different distance metrics, including Manhattan Hamming. Well, to summarize, the best area under curve was demonstrated by neural network. So then we were interested in the ranking of attributes. In our case, the attributes are single nucleotide polymorphism. And in the case of logistic regression, we estimated the so-called Shapley value and compared the SNPs by Shapley value. And on the left graph, we see the Shapley values for the top 20 SNPs for all chromosomes from 1 to 22. And we were surprised when we saw that the leader is the same SNP that was identified by genome-wide association study in our research. And if you look at the chromosome 4, the same SNP on the top of the list of that is ranked by Shapley value. This is very interesting that we see the same SNPs identified by two different approaches, GWAS and machine learning. And speaking about the second SNP that was found with GWAS, it also has a certain Shapley value, but not just very extraordinary, but at least at present in the top 20 list. So, to summarize the results of this research, I would like to tell the following. So, we detected two new single nucleotide polymorphisms associated to ischemic stroke. And both are confirmed at least by two protocols. And they both SNPs can potentially influence on the following genes, UNC5C and VWC2. And that's interesting that UNC5C is associated to Alzheimer's disease, and one of the SNPs identified, namely RC7796855, is associated to Parkinson's disease. They are probably the common molecular mechanisms of development of neurodegenerative diseases and ischemic stroke. And in this research we also tested 6 machine learning classifiers, and the best classifier was neural network. It showed the area under receiver operating characteristic curve being equal 0.69. And RS7699716 was detected by GWAS and logistic regression. I would like to thank you, the doctors from Pirogov Russian National Research Medical University, Elizaveta Petrova and Evgenia Koltsova, for the creation of collection of blood samples from ischemic stroke patients. The study was funded by Russian Foundation for Basic Research. Thank you very much for your attention. If you have any questions, proposals, feel free to drop me email message.